post co-evolution.
Thanks, Misha, and thank all the organizers, of course, for organizing this wonderful meeting. It's been a lot of fun so far. And so today I'm going to tell you about some theoretical work that we did along with Alexandra Walczak and Michael Lessig, and much of the work was done, especially the simulations, by Jacopo Marchi. And this is pure theory, and it's about coevolution of viruses in immune systems. And so even though it's theory, it's inspired by some data, and so this is the way that, for instance, influenza evolution looks like. If you look at H3N2, it looks like fairly linear, right? You have a trunk that dominates the evolution, and you have low diversity, which suggests strong selection on that strain. So that's one type of evolution, and sometimes you also have other examples. Sorry, my arrows don't work. Okay, such as flu B, which split into two separate lineages, the Yamagata lineage and the Victor lineage back in the 70s. And so we like to understand these different patterns of evolution using models. So what drives the evolution of these bugs? Well, it's mostly the immune system. And so one way to represent this evolution is also to see how these different strains along those tree are targeted by our immune system. So one way to do this is that to define some antigenic distance between different strains, and this is done by looking at the similarity of response to antibodies, basically, of different individuals, in this case, actually usually ferrets. And using this distance, you can do dimensionality reduction and project those strains onto a two-dimensional map, right? And what you can see here is that the evolution actually processes fairly linearly from the oldest one here to the newest one here. And what's quite kind of striking about this projection is that it seems to be always moving forward. So it's kind of a scorched earth policy, that it never seems to be coming back to where it was. And of course, the reason for this is because there's immunity in this region of antigenic space. And so the viruses tend to evolve towards regions which are, of the antigenic space that are not covered by immune systems. And that's what we think is moving the evolution forward. And these are more recent instantiations of this kind of two-dimensional maps on the 4 circulating strains of the flu currently. So we can look at this more precisely in terms of the immune system by putting both the immune system, so the antibodies in this case, and the strains, so which are represented by the antigens targeted by the antibody, in some sort of an abstract shape space. So the shape, if you like, is it's essentially the binding shape of the antibody to the epitope, but it's conceptually wider than this. It could also involve T cells and so on. It's just to represent specificity between different strains of the bug and different antibodies or immune specificities. So here in this space, you have a bunch of antibodies, right? And each antibody actually covers a portion of shape space, and that's called cross-reactivity, meaning that one antibody can actually recognize several strains. And then strains can be covered by one or several antibodies, right? And so in this idea, then a strain presenting certain antigens to antibody will try to evade immunity, will try to go away from where the antibodies actually stand. So what's the dimensionality of that space? If you think of the entire space of all possible antigens, it's probably fairly high. It was estimated to be from 5 to 8. But if you think about a particular bug, such as the flu, maybe it's a lower dimensional, hence the two-dimensional projections that people have been looking at. So our goal is to try to model this escape in this low-dimensional space. And I should mention that we're not the first ones to do this. There's some vast literature on the topic, and I'm just citing 3 recent papers trying to do this kind of modeling. And the kind of model that we propose is essentially, it's fairly simple. So we put and protections and viruses in this common space. And so if we take one host, for instance, I get infected by the flu. Inside that host, while it is being infected, the flu may actually acquire some mutations. And then it will transmit either the wild type or the mutated strain to the next person, right? And here things can happen. Either the host that gets exposed to the strain actually has a protection there. In that case, that host doesn't get infected, and then the virus just dies there. Or the second option is that the host actually does not have any protection around that, and the host gets sick, mounts an immune response, and therefore creates a new antibody, so a new immune protection here, and also then infects other people, right? And it goes on and on like this. So, so Jacopo Marchi, a graduate student, simulated this process. It's an engine model, and by simulating the process, he could recapitulate essentially the kind of evolutionary patterns I was showing in the very beginning, and we distinguish four different types of evolution, but it's really two. So one is what we call ballistic. So that means that the virus strain, the viral population in this antigenic space just moves forwards, right, as a straight line. And if you look at the lineages, the phylogenies that this creates, you see this very nice linear trunk. And this was already observed in an early version of this model. Then it can also, this trajectory can also bend, and it's what we call diffusive random walk. And the trunk looks the same, but in antigenic space, it looks a bit different. And then you can also have some splits, so meaning that two strains get far enough away apart from each other that they start feeling their own immune pressures. And in that case, you get either a transient or stable split. This is what's been observed in flu B, although it's not clear that this split in flu B will survive for very long because of COVID-19. The flu B has been doing fairly poorly, and one of them may actually die in the process. So what parameter ranges? What parameter range supports these different types of behavior? You can actually draw some sort of a phase diagram and can look at different quantities. Here I'm showing the average number of lineages as a function of mutation rates and the typical mutation germ size. And you can see a region where you get this processive linear evolution versus some other region of the space where you get these stable splits, for instance, right? So this was an engine-based model, but what I want to talk about today more in detail is an attempt to do some more theory and more analysis, to do more math on this kind of models. And to do this, we switched model a little bit, and we went to a more coarse-grained model. So in this coarse-grained model, we just represent the population of viruses as a density of viruses, which we call N, in this antigenic space, right? And each virus diffuses in this antigenic space. So that's to model the effect of mutations with diffusivity D, right? And as viruses infect hosts, they leave behind immune protections in host, and these will be represented by density of immune memories H, represented here by these antibodies. Since each antibody here can actually protect over some range, which is called the cross-reactivity range, they will actually, in the immediate proximity, reduce the viral fitness, right? So the green stuff here means actually that the fitness, the immune coverage is high, and therefore the fitness will be low, right? And as a result, even though the viruses diffuse neutrally in principle, they will tend to grow faster when in the white region, and that will actually drive this population forward as it leaves this trail of immune memories. So mathematically, we get a growth equation just for the viral population. So you get growth, the effect of mutation, and demographic noise, which is important in this case. The immune system just follows the population of viruses, so it just grows also in proportion to how many viruses there are in that region of antigenic space. And this is just a normalization term, compensatory term for accounting for the fact that we have a finite population with a finite immune memory. So whenever we add a new immune memory, we need to remove an existing one. And these two things are coupled through the fitness. So the fitness, what does it look like? It's a function of the basic reproductive number, R0, and the immune coverage, C. 1 minus C to the power M is the probability that there's actually no immune coverage at that region, because we assume that each individual has M immune memories, right? And so the immune coverage is simply this, is essentially this green stuff. is the convolution of the density of memories with this cross-reactivity kernel that has a radius 1. So if we put these equations in, again, simulate them numerically, we can get the sort of solution that I was describing before in the agent-based model. You see the viral population moving forward and leaving in its trail this immune protections here in blue, right? And if you zoom in, you can define the properties of that wave, such as its velocity, its width, in both directions, the directions of motion and the direction perpendicular to motion. So, you see this is what we call an antigenic wave. So you can also call it a oscillatory wave, or in physics, soliton. And it will be very reminiscent of the fitness wave that we may have heard about, and that Michael Desai talked about extensively yesterday. So if you instead of showing the distribution in antigenic space, you look at the distribution of fitness, you find this sort of Gaussian-shaped distribution. And the idea of the fitness wave theory is that the guys here that are at the nose of the fitness distribution. So here in X, sorry, you don't see here, but in X here is the fitness. These guys grow faster than the guys at the tail. And because they grow faster, that will actually move the wave forward, right? So you can view this as a projection onto the x-axis of this distribution here, because the immune trail here creates a gradient fitness, which is a fitness gradient, which is shown by these lines here, that make it more advantageous by these few strains here than people in the bulk. And this is the nose of our antigenic wave, right? Which is the counterpart of the nose of the fitness wave. And so because of this fitness wave theory is well established, we can build upon existing results. So there are two differences with the usual fitness wave theory. One of them is that even though I said that this fitness wave here moves in fitness space, is not really true, because it moves with this speed. By the same time, the immune system is catching up, and therefore the fitness of the entire population also goes down. And these two things compensate each other. It's as if you were walking on a treadmill. It's called a Red Queen effect, and this has been already noted in similar models. And so that's a slight difference with the fitness wave model. Another difference is that population size here is not determined. It's not a parameter of the model, and it's actually regulated by the immune pressure. So to illustrate how that works, I'm just projected here onto one-dimensional, the antigenic coordinate, this wave. So you have the viral population density n, and then the immune protection density h. And this immune protection will create this fitness gradient. So now imagine that you increase your viral population by some amount. What this will do is that later on, and sometime delta t later, the wave will move, but then the immune pressure will also be higher behind. And if the immune pressure is higher, the fitness is lower, and the fitness is lower, then this will actually decrease the population size. So that actually makes that the population size eventually converges to a fixed point, so a set point. And this fixed point is given by this fairly simple equation that gives you the ratio of the population size. So this is the number of infected hosts n divided by the total number of hosts nh. So it's also called the incidence rate. It's just given by the speed of the wave times some coefficient s, and this coefficient s is just basically the fitness gradient. So it's the value of the fitness gradient, and you can calculate it explicitly as the parameter of the model, as a function of the parameters of the model. So that relates n with v, but what is the speed of that wave? And this is where we can actually use existing results from the fitness wave theory. So it's given by this formula, and you can see here that it's a function of the fitness gradient, it's a function also of the diffusion coefficient, so that how much the virus is mutating, but also is dependent on the population size itself, because since this fitness wave is driven by its nose, so it's the guys at the front of the wave that grow faster, these are outliers, and how far these outliers are from the bulk depends on the population size, and this is where this dependency comes from. So if you combine these two equations, this one and this one, you have two unknowns n and v. You can actually determine these two parameters of the wave, and then you can go and compare with the theory, and we find that it works actually fairly well. So we just solve these two equations, then compare directly to the simulations, and get good agreements. Now we can also predict the width of that wave in the direction of motion. And to do this, actually, all we have to do is use a Fisher's theorem, because basically the position along that antigenic position is essentially proportional to fitness, if you assume a linear fitness gradient. So we just get a coefficient S. And so Fisher's theorem relates the width of the wave to the speed of the wave in fitness space, and you can convert that into antigenic space, and you get this formula. And plugging this in this formula, you can actually show that this works. It's not maybe that surprising, but it's a nice check. We can do something even better, which is that we can also predict the width in the perpendicular direction. And the prediction actually is that this perpendicular direction is square root of 1.66 of the parallel direction. So where does this, I mean, the fact that it scales linearly maybe is not surprising, but where does this thing come from? So to see how we can predict this, we imagine two strains of the bug, and we ask how far they are away in this perpendicular direction, and the answer is just given by diffusion, because in that direction things are completely neutral, and so all we have to do is multiply the diffusion coefficient by twice the T2, so the time to the common ancestor. And what is T2? It's given actually by the fitness theory as scaling like sigma square over d, with this coefficient, which was measured numerically in this work. And so by plugging it back in, you get this nice linear relation. Okay, so now we can go even slightly further. So now we have essentially an effective description where we have this fitness wave that moves in the direction of the fitness gradient. So we can view it as a point-like particle moving with this speed. So following the gradient plus some genetic drift. And so why is that genetic drift? It's because since the wave is driven by the fitness nose, the few individuals at the nose still drift in the perpendicular direction, and that will actually have an effect. So I say it follows the fitness gradient. I mean, formally, actually, it's the same thing as the gradient of coverage, because fitness is just a nonlinear function of coverage. And this coverage is simply given by an integration of the past positions of the virus itself, right? And so this is basically encoding this trail left by the virus. So what that means is that if you didn't have this genetic drift, then the wave would always proceed forward, because basically it's following the gradient of something that only depends on its past position. So it should continue going straight. So that actually creates some sort of an effective inertia. And genetic drift would actually slightly deviate this inertia, slightly deviate this straight motion, and in the following way. So if the wave is moving forward, because this nose tip strain actually diffusing a little bit, this will actually deviate slightly this motion, and effectively the wave will the direction of the wave will undergo some angular diffusion, and we can define some persistence time that defines how long it takes for the wave to actually change direction significantly. And so we can actually write an effective equation for the diffusion of that direction. It's only valid for fairly large times, and from that we can actually get an expression for the persistence time, so the time it takes for this trajectory to bend. And in the end, the results only depends fairly simply on the model parameters. And in particular, so this is like a constant prefactor. Here you have to imagine that D is very small, right? Because mutation rate is usually very small in a single strain. So this T persists, this time persistence time okay, so we can also check that it agrees well with the simulations. So this persistence time, this is essentially proportional to 1/d, and 1/d is essentially the time it takes for a single virus strain under no selection, just neutrally, to escape immunity. That is to walk by a distance 1 in antigenic space. I remember distance 1 in antigenic space was the cross-reactivity radius, right? And this has to be contrasted with the time it takes for the whole population to escape immunity. That's given by 1/v. Sorry. And that actually scales more like 1/d to the power 2/3 minus this logarithmic corrections. So I'm gonna wrap up. I think I'm quite on time. So we've proposed an analytical theory of coevolution in phenotypic space. So, suppose this kind of solution, and we have analytical predictions for the speed and other parameters of the wave. And we show that the speed, maybe not surprisingly, but we have an explicit scaling is much faster than it would take the virus to escape just by itself. So we get a collective population effect here, right? So the fact that you have a large population allows you to escape immunity faster. We also have a prediction for how often or how fast this antigenic wave changes its angle, right? And that is actually given by the time for a single virus to escape immunity. And something I didn't talk about is that we also observe these splits in these models, and we're working on some analytical theory to predict the rates of these splits to sort of know how often should we expect, for instance, the flu to split again in sublineages. And this question was already addressed in a slightly different model by these authors. And to finish, I mean, I talked about the flu as an example and maybe viral solution, but I think this kind of formalism is quite general, and it can also apply to many other problems of coevolution. So you can think of phage in bacteria. Of course, that's also a problem of immune host coevolution, but maybe also predator-prey problems living in some common phenotypic space. And yeah, so with that, I thank you for your attention, and I'm happy to take questions.
Thank you, Thierry. While people are adding questions to the chat, something I was wondering, could you comment on, like, maybe you mentioned this and I missed it, but the dependence of this behavior on the dimensionality of the trait space?
Right. I didn't talk about it actually. So, but like many things are actually insensitive to that. I mean, most things are insensitive to that. So this diffusion, for instance, you can put any dimension here. It won't change the results. Like whenever you have a result for, you may multiply by the dimension for, but like the width, the speed, all these parameters, the speed, the width in both directions, all this is true in any dimension.
And is that surprising at all? Like I would imagine, just the usual properties of diffusion depend strongly on dimension. In 1D, it would be sort of persistence in one direction necessarily. But you,
in 1D,
naively would have imagined that it can.
Yeah, no. So that's a good point, and the answer is that it doesn't. Of course, if it's in 1D, then you don't allow for any drift in the other directions. But once you say that, then once you put D larger than 2, 2 or larger, then it doesn't really matter. Like each other direction is basically independent of each other, of the first one. You do get a spontaneous symmetry breaking, which is that the wave has one direction, right? And then all the other ones are like perpendicular fluctuations.
Okay, thank you. Okay, let me read out some questions from the chat. One is, actually also about dimension. So there's what sets the dimension of phenotype space? And do you expect some level of generality for high dimensions? Is this a numerically or analytically feasible regime?
Right. Okay. So what sets the dimension of phenotype space? I think that's a more an experimental question. And so I don't really know. I mean, I think for the flu, I think maybe there are people who are more experts. I don't really know. I've seen them people projected in 2D, but I couldn't really decide whether they projected into 2D because it was easier to represent, or whether you could provably show that 2D is enough and that 3D doesn't add anything. So I'm not really sure. Do I expect some generality for high dimension? I already answered that. And is this numerically feasible? Probably the answer is no. I mean, numerically it's very hard to do in much higher dimensions. Analytically, as I said, it doesn't really change anything. I hope that answers the question. I don't know if Aditya wants to add anything.
No, thank you. Yeah, I think that answered the question.
Cool. Then there's a question from Eric. Is it possible for the virus to go extinct in this theory? And if so, what determines the chance of this happening?
Yeah, absolutely. It goes extinct. And actually in the simulations, it happens all the time. So we sort of condition on it not going extinct, but things are fairly unstable. And you could also try to estimate the rate of extinctions, but that's pretty hard. So we haven't really explored that very much, but it's a very good question. There's something that always confuses me is that if in real epidemics, things never go extinct. I mean, not really, right? For instance, like you look at the flu right now, it has an R0 way below 1 and so on, but it doesn't actually go extinct because there's always a reservoir. So you always have people who have it for a long time and so on. And so things never really go extinct for real, right?
Yeah, no, the reason I'm asking is sort of previous work. I think a lot of people worried about how you get things to persist for long times and not make splits, so that, whether you can get the model to have enough of a parameter regime where you don't get lots of splits.
Yeah, no,
so I mean, don't go extinct all the time.
Yeah, no, I'm aware that's a big problem, and we sort of try to avoid that problem. I mean, not avoid the problem. I think we try to look at the different thing. Right? We were not looking so much at the stability. We were interested in the splits, but there's a wide range of parameter regimes where things are fairly stable, especially when you get a nice straight line, then it's very stable. There's another problem, which is explosion. So when things, like pandemic, that also happens in the simulations. So that because here, like, we always want incidence rates that are fairly low, right? But sometimes the simulation blows up and the virus just escapes in a place where nobody has any immunity at all, and then everybody gets infected immediately. And so that's not good for the sake of our simulations, because we were thinking in terms of steady states, right? So I think that there is a good question to be asked about sensitivity to initial conditions in the case of an outbreak. What happens with extinctions? I mean, extinctions, as I said, I think no model is ever going to be really realistic when it comes to that. Yeah, but the splits, we do have splits in this. We can quantify them. It's just like we haven't quite finished doing the theory about it, but I think it's gonna come.
All right, thanks.
And I should mention in this paper by Jan Neher and Scheiman, they looked at a slightly different model where they were not in phenotypic space. They were in genetic space, so infinite dimension, where any mutation is an escape mutation. And in that case, they had a scaling theory for the rate of splits and also for the rate of extinctions. Okay. So yeah, people have already looked at this question.
Yeah, right. Got a question.
Oh, that's on.
If we can, okay. Maybe I missed it, but what is the rationalization for the population? A simple explanation for the fact that population doesn't spread laterally, so it stays compact in the lateral direction.
Sorry. In the lateral, it doesn't spread in the lateral direction. So okay, so that I mean, thank you. Maybe that means I didn't explain this well. So it's because of this. It's because you get selective sweeps all the time. So everybody in the population is somewhat tethered. They're all offspring of some guy who used to be at the nose, right? And they used to be at the nose at the time T2, which is which is equal to the distance between the bulk and the nose divided by v. And that's essentially proportional to, that's Tn, right? That's the coalescence time. But it's actually the same as the pairwise coalescence time, right? And this pairwise coalescence time, therefore, is finite. And because of that, it cannot spread for infinity. So that's what determines the sigma. And I think it was a bit of a surprise. I mean, I didn't know what to expect, but I expected maybe this width here would be square root or some power of the width in this direction, or maybe some combination with the speed. But it turns out it scales exactly like the speed in the direction of the wave, which didn't have to be the case. I mean, maybe there are arguments for why it has to be the case, but to me, it wasn't completely obvious.
Thank you. And Benny, I saw you unmuted. Did you have a question?
Yeah, I just wondered whether you thought the model could be extended to sort of faster systems like in-host evolution of HIV, where the rates are much faster of mutation, and selection is
that's a great question. I think it would work there as well. I mean, the timescale would be different because here we're thinking about hundreds of years, right? So when you think about the flu, so to give some sort of a timescale, right? So when I talk about the speed and the turnover rate of the flu strains, that's about 10 years, right? A few years, let's say, maybe more. But like the time it takes for the entire population to renew its immune memory is about 10 years. Yeah. People get infected by the flu every 10 years. So the prediction we make here is that the time it would take for this nice antigenic wave to actually bend, which we haven't really seen if you look at those antigenic maps, it's not really clear it happened yet, would be more of the order of hundreds of years. Right. So like we're talking about very different times. In-host HIV immune system coevolution, maybe you can actually write the same theory, right? Because like this kind of theories, this kind of model here, it would work the same, right? It's just here we summarized as if just one person. And so you could imagine having some similar theory as this one, but you would just interpret the timescales very differently. So for instance, here for the flu, the typical timescale was like infection cycle. So about 3-5 days, maybe a week. But for HIV, it's I guess, well, I don't know, actually, you tell me. It would be more like the reproductive number. So the growth rate of HIV inside the host.
Yeah.
But then, as you said, the mutation rates would be much higher. So I don't know. It's a good question. I haven't really tried to plug in the numbers.
I think you get much less clean. I mean, you tend to get multiple strains by the end of the infection quite often.
Like there's a regime here in which everything sort of explodes. I mean, meaning that's then you get high diversity and you don't really get a wave because things really go in all directions. And I think it is an interesting question of what's going to happen for SARS-CoV-2 because it's an outbreak. So at the beginning we haven't reached any kind of steady state yet at all. And since it's an outbreak, like we see new variants coming in, but like each coming maybe they all seem to be affecting the same region, right? But like it could very well be that different variants would find different ways to escape, and this could actually create sublineages potentially. But I don't think the theory can answer that really because it depends on the molecular details. But it does tell you there's a possibility.
Thank you. I'm sorry to cut this very interesting discussion. I think we should move on to the next speaker, and we can